This is Jose Merino, Editor-in-Chief of the Neurology family of journals. The Neurology podcast provides practical information to neurologists and other clinicians to help them provide better care for their patients. Thanks for listening and have a great week.
Hi, this is Stacey Clardy from the Salt Lake City VA and the University of Utah. Today I'm joined by Walter Koroshetz, Director of the National Institute of Neurological Disorders and Stroke, NINDS. He's here to update us on what's new in patient-impacting developments in neuroscience, current research priorities, and how our collective neuroscience and neurology community can stay creative amid funding, policy shifts, and all the rapid changes in the field. This is going to be a two-part series. We have so much to discuss on the 75th anniversary of NINDS in terms of developments, the past, the present, the future, where things are. Walter, thank you for being here today.
It's a pleasure, Stacey.
I have so many questions. Let's start with some wins for patients from the past couple of years since we last spoke. What recent NINDS or NIH-supported advances would you highlight as practice relevant right now? And I'll tell you, I have a couple favorites, and maybe you could speak to them too. I'm thinking especially of the adaptive deep brain stimulation for Parkinson's disease, which just saw FDA clearance and uptake. But you tell me, what are your favorites and what should clinicians know about what's changing quickly because of the research?
Thanks for asking, Stacey. I start off by saying I love all our advances equally. I don't have any favorites, but you brought up a really good area to talk about, and that is neuromodulation, modulation of neural circuits. As trained as a neurologist, we see the patient, we listen to their story, we do an exam, and we make a judgment about what circuits in the brain are affected to cause the symptoms and the signs. Sometimes we look at the imaging. Sometimes the imaging shows you something that's consistent. Sometimes you don't see anything on the imaging. And that's basically how we practice based on our understanding of neuroanatomy. But the big black box forever in neurology has been we don't have a way of measuring the outputs of those circuits in any objective way. If you think about our technologies to look at circuit function, EEG is really the one that's been clinically the most useful, and that's a technology from 1928, if I remember right. So this has been a big black box all these years. And when you think about the brain, from the neuroscience standpoint, the brain is an information processing machine. There are all these circuits, and they are not really confined like we thought they were into the homunculus. So if you look at a brain now with the technologies we have, the entire brain becomes active in almost any type of behavior. And this activity is millions and millions of neurons activating in some kind of sequence, and we have no idea how to understand or interpret what the language of the brain is right now. We know about action potentials, and we know that one cell can excite another, but how do you deal with a circuit where you have millions of neurons and you know that their activity is what is causing a behavior, directly causing a behavior, or directly related to a sensation? In the past, people would study the brain by putting electrodes and measure the activity of one or two neurons at a time in a awake behaving monkey. But now you can see a million neurons simultaneously in awake behaving mouse. So the shift has gone from not able to get the data to now you have so much data. How do you really understand the underlying principles of how that activity is processed? So that's really the big change. And that all came out of the Brain Initiative, which is a really well-funded project over the last 10 years to develop the tools to look at circuits. And that has led to, as you mentioned yourself, the idea of adaptive brain neuromodulation. So the idea here is that you can detect signals in the brain itself that determine how you want to modulate the circuit. Early on, it's finding an oscillation that's associated with bradykinesia in Parkinson's, stimulating to make that go away. So it's adaptive. It's closed loop. All the changes are being made based on what's being measured inside the brain, and it's happening in real time. And that's what was recently approved in Parkinson's, but I would say that similar projects are going on in other disorders, epilepsy. We just actually today saw a whole bunch of projects that are coming in the epilepsy space and the Parkinson's space related to this, but also in things like depression, obsessive compulsive disorder, chronic pain. There's a signature of chronic pain that's been identified in some patients that is then ameliorated by stimulation. So this opens up a whole new way of approaching symptoms in our patients for benefit. And it all comes out of the brain initiative technologies. The only thing I'd say to close here is that the stimulation parameters that we're using are very crude. They are basically electrical stimulation. You're sending electricity in the brain. It's not entirely clear what that's doing. There are much more precise ways of manipulating circuits that are becoming, and these are going to be cell type specific ways of modulating circuits as opposed to geographic location wise stimulation.
It's all very exciting. So already new targets, more precise targets, but as you said, an entire paradigm shift away from the electrical stimulation is coming close on its heels. So that's really exciting. Another topic that always catches my eye, and that's rare disease. So I was just reading that NINDS launched what's called the ultra rare gene-based therapy or URGENT network to move some tailor-made treatments into first-in-human studies. So what is this going to look like? What are we going to see happening? What is unique about this URGENT network that will allow for faster solutions here?
We have a responsibility to get these genomic therapies out to people as fast as we can, because as we have so many neurogenetic disorders, many of them would really threaten life, cause death in young children, or permanent disability, affect a person's ability to lead a fulfilling life. And so there's a real urgency here. So that's why we named it URGENT, because of the urgency. But the other thing which is important to understand is that when we were developing drugs, the average time from a discovery to a drug hitting the market is about 20 years. And that was tough to overcome. Things went into the pharmaceutical companies. They had to do a lot of testing, manipulations, get their binding right, do the formulations. And it just took a long time. But for genomic therapies, you can make them in your lab, right? There's no IP on the mutation. Developing a gene replacement can be done in the laboratory. Developing an antisense oligonucleotide that blocks production of a gain-of-function mutant protein can be done in the laboratory in a matter of weeks to maybe even shorter. So the question becomes tinkering with the genomic therapy, but the actual sequence is not going to be that hard to figure out. It becomes a matter of delivery mechanisms. But if you have one delivery mechanism that works, the beauty of genomic therapies is you just use that Trojan horse and just change the genomic therapy for the disease that you want. So once you have the delivery mechanism, you should be able to move very quickly from one neurogenetic disorder to another, potentially actually go after multiple at once. And so that's what I think is what we have to recognize going forward, that there is urgency because these diseases are so terrible for folks. And we don't really have a good excuse why, for instance, in spinal muscular atrophy, we have a gene therapy that prevents death in young infants due to motor neuron disease, and it works. So if I have another neurogenic disease, what's my excuse why my disease doesn't get that same therapy? And we don't really have a good excuse anymore. We really should be able to move things very quickly. And the technologies to do that, as I said, in terms of delivery, they look real promising that we are going to have, say, AV virus vectors that get into the brain very potently. And so then you have your delivery mechanism. Then you should be able to just move from one disorder to another very quickly. Now, that being said, it's not as simple as that. There are certainly some dangers. There are things that we don't know that we're going to find out along the way. This area of genomic therapy, we mentioned ASOs, gene replacement. The other area where I'm spending a lot of time is in the NIH genome editing program, and that is absolutely amazing. So the idea that you can find a mutation and correct that mutation and cure a child is just enormous. And there was a great example of one of the programs we fund published in New England Journal where they found a mutation in a child. Within 6 months, they developed the therapy, got FDA approval, got money from us, and treated the child. And so 6 months, where, as I said before, for the drugs, it's 20 years. We're in a really different spot right now because we have the ability to develop these therapies very quickly. They're potentially much more potent than any drug. I used the elevator pitch that drugs are stupid. They don't know where to go. They go everywhere. When you don't want places, you get a lot of places that you don't want them to go, and they go there anyway. But these genomic therapies, we have ways to hook them up to cell-specific enhancers or promoters, so they just go exactly where you want. There's now epigenetic editing, so you can basically decrease or increase the production of pretty much any protein you want. So the toolbox is so much more powerful in the genomic space. Yeah.
Wow, that's really exciting. So what is the major barrier there? Is it money? Is it manpower? What is it that is giving you the most headwind?
On the academic side, things are rolling pretty quickly. It's a matter of there's a very few places that have the expertise to do this. So expanding across universities, having more universities get into genomic therapies would be really fantastic. But the other problem is the commercialization side. So remember, for any treatment to get to patients in our world, it's got to be taken up by a company. Now, whether or not that model works for genomic therapies is a question. Certainly for the really rare diseases, until there's a company that has a platform that can treat 100 rare diseases, companies are not going to go there. So what we really need is a mechanism to move from the academic labs where the therapies now can be made to a government, university, potentially biotech partnership where they can be tested in people, and then to some type of a nonprofit group that can, once these have been demonstrated beneficial, can make them available at cost. Because that's the thing that's a little bit different. Remember, in a drug company, it takes 20 years. You need to make a lot of money to be able to resource all the work that went into that new drug. But if you can make something in 6 months, the hope is that you can make that available without a major profit. And if it's a nonprofit who's running it, they could do it at cost and not have to worry about making $1 gazillion for their stockholders. So there's an interesting paper by Jennifer Doudna, who was one of the discoverers of CRISPR gene editing, about a new model for making gene editing therapies available. And it's a very different model than what we have now for drugs. It will take some effort and a paradigm shift, particularly on the university sides, to basically allow their IP to move in this direction of a nonprofit that makes things available for cost as opposed to trying to sell things for multi-million dollars to a company. So anyway, that's my spiel on this.
It's useful. These are the real challenges, right? It's relevant.
We'll see what happens. But I think that's the biggest barrier is getting to commercialization. So NIH, a common fund, we can show that it works, but how do we sustain it over time? We really need some partners in terms of getting these therapies out to people and doing it so that the price is not outrageous.
Yeah, partnership really being the key word there, creative thinking and partnership, and how do we get the win for these patients with all of the resources from the different partners? Yeah, it's a great point. I could talk about rare disease all day long and the challenges there, but how they inform ultimately how we do research too. But speaking of maybe not as rare as we would like it to be of a disease, talk to me about long COVID, right? This continues to trouble patients. Certainly, most neurologists will see some of these patients in their clinics. So give me an update from your vantage point on the RECOVER study. What has it clarified most recently in 2025 about where we should be heading here? Phenotypes, mechanisms, and trial directions related to long COVID.
It's different now than it was 2 years ago or 3 years ago. When COVID first happened, People were in intensive care units. Everybody knew you developed ARDS. It's going to take you years to recover. So people set up post-COVID clinics. And the big surprise was that these clinics were filling with people who were never even hospitalized. But years have gone by, and now it's different. A large percentage of people have recovered. So our focus now has to be on the people who did not recover. So there's a lot of interest in understanding why you get long COVID, what the risk factors for long COVID are. But I think now the shift has to be, there are people who 3 years later are still not better. The take-home point is that those people who are not better 3 years later, they have a condition which really fits what's called myalgic encephalomyelitis chronic fatigue syndrome, MECFS. And that everyone suspected was a post-infectious chronic condition, but no one knew what the infection was and no one could prove it. In the COVID space, it's pretty clear it was related to COVID. So that's really the mystery. What happens to people who have an infection, and it may not be something that puts you in the hospital, but it causes this chronic fatigue, pain, trouble sleeping, trouble with memory, and this very weird thing called post-exertional malaise, which is defined as, just in layman's term, you may not be feeling good one day and next day you might feel a little better, and you go to the store, go out, walk a mile to the store and come back, and then you're basically wiped out for 3 days. Something happens during the exercise that just makes everything worse for a period of time, which could be 3 days. Some people might be 3 weeks. And that's the really peculiar signature of MECFS. And it's also, turns out, to be the single symptom in long COVID that predisposes people to a chronic condition, a severe chronic condition. Those are the most severely affected. The most severely affected are the ones who, unfortunately, have the less chance of recovering over time. So we're really into that mystery of what is this post-infectious condition. And of course, as neurologists, we have to think about what is the neurobiology of fatigue. Fatigue is very common, but it's not something that comes from the muscles. It's a brain state. And we have to really get in and figure out what that brain state is all about. There's lots of things that can cause that brain state, of course. Any infection, there's lots of drugs. You get into it with people with MS, traumatic brain injury, post-stroke. It's not uncommon at all, but we don't have a clue of what that brain state is all about. And then, of course, there's now with long COVID and the Recover program, we've enrolled 15,000 people into this amazing cohort study. We have the samples, we have the patients to try and get to the bottom of it. I always say that this is really the last great medical mystery of all the conditions that affect people, certainly large numbers of people, where there's very little to no understanding. This is it. And so it's a real challenge to understand it. Recover is our best chance at getting there, but I must say, after 3 years, there's lots of culprits, but no one knows who's really guilty yet in terms of the cause. The cohort is being followed up. There are samples, thousands of samples. There are studies that are still going on in the patients who have the symptoms. Depending on their symptoms, the studies are customized. And we have adults, we have children, and we have pregnant women cohorts. There's autopsy studies to try and see if people who die of whatever reason, who have long COVID, whether we can see something under the microscope that it's just not going to be able to get at if you just start looking at blood samples. That's ongoing. And then there are a large number of groups around the country who have been funded to try and understand the pathophysiology of long COVID. There are clinical trials that are ongoing. Some of them are concluding soon. And there's a new set that's going to be launched actually September 9th. There's a meeting here in Bethesda of people coming in to pitch ideas on the next trials for long COVID. It's called Recover TLC. It's the next phase of trial. So yeah, the work is ongoing. We've been working, and people may not know this, but Dr. Francis Collins asked NINDS, probably 10 years ago to really come up and take hold of ME/CFS research along with the Infectious Disease NIAID Institute. So we've been working with them on ME/CFS for years before COVID happened. And we're really good partners with them now, along with the Heart, Lung, and Blood Institute on Recover.
Yeah, in science and medicine, we always look for every opportunity to turn a tragedy into something better for the future. And this is the perfect example. When I'm talking to these patients, I often say, look, in neurology clinics, we knew about this long before COVID because we've always seen patients who, whether identified or unidentified, have a virus. And we've seen this with a multitude of viruses. And then what COVID brought us was a high number of individuals, unfortunately, but a high number where we knew the culprit, right? So they could be studied in a systematic way. So I'm glad to hear that there's still focus, there's still progress.
If you talk to people who have this condition, they had a really hard time finding doctors. A lot of doctors just didn't believe the patient. I'm not sure how you cannot believe a patient, but they just felt frustrated. They didn't know what it was, and they just assumed it was some psychological problem the patient was having. And that's very frustrating to people who are debilitated like that. The one silver lining of long COVID is that, as you said, we had this pandemic and now there's hundreds of thousands of people with long COVID. Pretty hard to brush that under the rug anymore, that's for sure. So hopefully more physicians will really seriously take the pains taking care of these folks now.
Yeah, I always joke with the patients that it hurts our egos when we can't figure something out with a clear answer. And so I think sometimes the patients sense our frustration that we don't have an easy answer. But hopefully, as you point out, one thing that we've learned is the importance of a multidisciplinary approach for these individuals and continuing to give hope because the research is ongoing. And I'm glad to hear there's a new round of trials coming up too. I guess along those lines, what are your top NINDS priorities going into 2026? And how should investigators, the researchers, the clinicians align their proposals, whether they're neurodegeneration, circuits, systems, device, therapeutic translation, all the things we've been just briefly scratching the surface on so far? How should investigators align those proposals going into the new year?
I would counter and say that what an investigator needs to do is to come to a sense of what they really want to do, what they really want to accomplish, and then just figure out how to get somebody to pay for it. And
easier said than done.
Easier said than done. But I think you really want to stick to your guns in terms of what you really want to do. Life is too short to do things that you don't want to do because of some fuzzy stuff.
Fair. With that in mind, though, with your bird's eye view, let's think about the young investigator who maybe got their graduate work completed or did a project and now they have the tools to conduct scientific research, but it's overwhelming, right? The rate at which things are moving. So maybe I ask it differently. Maybe I say a young Dr. Walter Koroshetz comes out of training, has several things that he's passionate about. How does he view the future? How does he view where technologies are moving? How does he focus that energy?
I think everybody does it differently, for sure. I think one rule that's helped me is to have a couple of things that I was really interested in. And truth of the matter is, some of them looked really good for a while and then sputtered, and others looked good and then exploded, and some never really got off the table. If there's only one thing you want to do in your life and you really narrow it down to one thing, you're putting yourself at risk. If you have as your goal in life a broader goal in which there's many ways to get to that goal, I think you're better off. The beauty of neurology is that we're in this ecosystem with the patients, really at the ground level with the patients. And that allows us to bide our time also and look for opportunities as they pop up. And neurology is moving so quickly in terms of certainly in the new areas of therapeutics and diagnostics, that if you stay in the game, it's almost impossible not to be able to make a major contribution. That's my sense. Some of the other fields I look at, I wonder, boy, they know so much about diabetes or coronary artery disease. how would you really make a difference? But in neurology, that's not even a question. There are so many things to do. As I said, you have your goals in mind and see how you can find somebody to pay for the work that needs to be done. And that also should be diversified. The great thing about neurology is that there are disease organizations, there's the academy that funds projects, and NIH, I think, just to state the obvious when people are reading the papers, is that there's a lot of uncertainty about the near-term funding situations at NIH. We don't know what our budget is going to be. People may not know this, but we never know what our budget's going to be year to year. Our budget is given to us by Congress on an annual basis. So it's not unusual for us to be in a kind of ambiguous situation, particularly now, which is coming to the end of our fiscal year. Our fiscal year ends at the end of September. Now, looking forward, we have a couple of options that we're looking at. One is what's called a continuing resolution, which means we have the same amount of money to work with that we had last year. And the other is the bills that the House and the Senate have to compromise on. The Senate bill basically is really good to NIH. It keeps the funding, actually gives NINDS a little increase in funding, a little bit of increase to NIH in general. We do every year. We have to wait and see what the Senate and the House compromise on going forward. But NIH has always gone up and down its funding amounts over the years. There have been good years, bad years. It oscillates. So that's why I tell people, don't fret about a dip, because there's always going to be oscillations. If there's a dip, there's going to be a rise. You just have to work a long-term plan, not get all anxious about stuff that's just short-term.
We'll stop there for today's episode. We are going to come back with a second part with Walter Koroshetz, where we discuss some of the challenges that are very real to all of us right now, strategies for addressing them, and continuing the important work of NINDS now in its 75th year. This is Stacey Clardy, your podcast editor. If you've enjoyed the podcast, please take a few moments to subscribe, rate, and review the Neurology Podcast through Apple Podcasts, Google Podcasts, Spotify, or wherever you listen. And remember, you can always head to neurology.org/podcast for our full list of past episodes. Or you can also search by keyword in your podcast app for any neurology-specific topics you want to learn about.